Good morning, everybody. As we finished gastrointestinal tract, the next chapter is about excretory system, which is also called the renal system, wherein we will be learning about anatomy of the excretory system, the blood supply, the functions of the kidney, which can be excretory and non-excretory function, nephron, the basic unit of kidney, which is the nephron. Should be knowing the functional and the basic structure with types of nephron, which is cortical and juxtamedullary nephron. As you all know, the kidney has innumerable functions, which is the formation of urine, which compasses the three steps: the glomerular filtration rate, which is called as GFR, the tubular reabsorption and tubular secretion. The second is the concentrating ability of urine, that is based on countercurrent mechanism, how water reabsorption takes place. The third one is micturition, the acid-base balance in the body, how the acid is and the bases are balanced. We have two types, the volatile and non-volatile acids. Then finally we have how the urine is excreted, that is the voiding of urine called as micturition. So today's class is about, the objectives are, what is the functional anatomy of with functions. As you all know, the kidney is placed retroperitoneally, produces urine, it transports downwards towards the bladder where it is stored temporarily, and it is voided through the urethra through the process of micturition. Kidney, you all know, is outside the peritoneum, so it is retroperitoneal. It is 150 grams, it's weighing around 150 grams. Kidney is not kidney shaped, it is bean shaped, lying retroperitoneal in the position of superior lumbar region and extend from the 12th thoracic to 3rd lumbar vertebrae. The exit is through the ureter, and we need to know it has supplied by the renal blood vessels, the renal artery, and the lymphatics and the nerves which enter and exit at the ileum. So this is the basic thing which has the kidney with the renal capsule, and we have the renal artery, the renal vein, which is coming out of the hilus, and that is the first branch of aorta. So we have the cortex and the medulla. The cortex is pale because of the sluggish blood flow, whereas the medulla, which is involved in countercurrent mechanism, is pink in color, which maintains the medullary hyperosmolality. You can see how the cortex goes into the renal pyramid. We have around 6 to 7 renal pyramids, which drains into the minor calyces and finally into major calyces into the pelvis. We have the renal papillae and finally into the ureter. So we need to know there is a renal lobe, that is what with major and minor calyces, and there is a renal pyramid. So it opens into the hilum and finally into the ureter. There is around 1200 ml of the systemic cardiac output which flows into kidney every minute. So how much is the renal blood flow? It's around 1/4 of the cardiac output, that is 25%, which is 1200 ml. The arterial blood flow flows into the kidney and the venous comes out through the hilum. The nerve supply, which is the innervation, is through the renal plexus. We can see how the aorta divides into renal artery, it branches into segmental, the lobar, then interlobar, and it forms an arc which is called as arcuate artery, which opens into again interlobular, finally into the afferent arteriole and the glomerulus. Glomerulus is a tuft of capillaries. Around 15 to 20 capillaries form glomerulus, and finally drains into the efferent, the peritubular capillary with interlobular, the arcuate, the interlobar. Finally, we have the renal vein draining into inferior vena cava. So, what are the functions of the kidney? Whenever we talk, it is excretion of urine. So, there are excretory functions and non-excretory functions of the kidney. The removal of organic waste from the body fluids. It filters around 200 liters of blood daily, allowing toxins, metabolic waste, and excess ions are removed in the urine. So this is the excretory functions. So what are the non-excretory functions? It regulates the blood volume by adjusting the volume of water lost in urine. It regulates the blood pressure by releasing erythropoietin and renin, and it regulates plasma ion concentration in the form of sodium potassium chloride by controlling the quantities lost in urine. It also helps in synthesis of calcitriol by maintaining the calcium ion levels. It stabilizes the acid-base balance by controlling the loss of hydrogen ions and bicarbonate ions in the urine. It also has endocrine functions, like it has a glucogenic functions where glutamine is formed to release the ammonia, so which is endocrine in nature. It also conserves valuable nutrients by preventing excretion while excreting only the organic waste products. It assists liver to detoxify, and it is metabolic. So endocrine was, I made a mistake. Endocrine is producing erythropoietin. It helps a role in vitamin D actions by converting the vitamin D3 first in the liver and the kidney. And finally we have metabolic, that is gluconeogenic, which forms the glutamine releasing ammonia. So you can categorize the functions of kidney into excretory and non-excretory. Excretory is excretion of urine along with waste products, whereas non-excretory functions are, it can be metabolic, wherein or the general blood volume, pH, all those functions and maintenance of blood pressure. Then we have the endocrine functions, which plays a role in converting the forms the active form of vitamin D, that is the calcitriol, 1,2,5-hydroxycholecalciferol, and it is metabolic, forming the glutamine, that is gluconeogenic function. The nephrons are the structural and functional unit of the kidney. There are around 1.3 million nephrons in each kidney, which has an afferent and efferent arteriole opening into a glomerulus, then forms a Bowman's capsule, which has parietal and visceral layer, and it opens into the proximal convoluted tubule. There are two parts convoluta and parts tubular, and finally into the descending limb of Henle, the loop of Henle, ascending limb, which forms the distal convoluted tubule, comes in contact with the afferent arteriole, forming what is called as juxtaglomerular apparatus, which forms the renin, and this renin plays a role in conversion of erythropoietin to erythropoietinogen to erythropoietin. Angiotensinogen to angiotensin 1, which regulates the blood pressure. You all know angiotensin 2 is a potent vasoconstrictor. So we have the renal corpuscle, that is the Malpighian corpuscle, which is the glomerulus, which has 200 microns, and it invaginates into a tuft of capillaries into dilated. Finally it blinds ends to the Bowman's capsule. The renal tubular part, which is the PCT called as proximal convoluted tubule, the loop of Henle, the DCT and the collecting duct which drains into the pyramid. So you can see how the afferent arteriole, the glomerulus, the efferent arteriole. Then we have the peritubular capillaries in the cortical nephrons, whereas the vasorecta in cortico-medullary nephrons. How the whole function can be dealt in this diagram, where the glomerular filtration takes place, tubular reabsorption takes place. 90 to 95 is reabsorbed in the proximal convoluted, where the reabsorption takes place from the lumen into the blood vessel, whereas secretion from the blood vessel into the tubular lumen. And finally, the loop of Henle, which is concerned with countercurrent mechanism in the corticomedullary nephron by maintaining the hyperosmolality of the medullary interstitium. And finally, the distal convoluted tubule and the collecting duct plays a role in forming GG apparatus and water absorption. The renal corpuscle, which is the Bowman's capsule, which has an external parietal layer with the internal visceral layer, which has podocytes which terminate into foot processes. These are slits. The glomerular endothelium is fenestrated epithelium that allows solute-rich, virtually protein-free filtrate to pass from the blood into the glomerular capsule. These are filtration slits which opens between the foot processes that allow the filtrate to pass into the capsule. Normally depends on the charge and molecular weight. If it is more than 60,000, it doesn't allow it to get filtered. So the function of the Bowman's capsule is filtration. So you can see how the tuft of capillaries which is covered by the Bowman's capsule, and you can see how the blood vessel covers the whole of the renal tubule, that is the peritubular capillaries. And you can see the general pale and pink, the pink cortex and the pale medulla because of the sluggish blood flow. The medulla is placed why? Because of sluggish blood flow or slow blood flow. So you can see the filtering membrane which has the endothelium, the filtration slits and the podocytes. Depending on the size and charge of the molecule, it can be filtered. The renal tubule has total length of around 45 mm, of which 15 mm forms the proximal convoluted tubule with a diameter of 55 microns. It is composed of cuboidal cells with numerous microvilli, that is, it helps in reabsorption forming, and it is enriched with mitochondria. This brush border helps in reabsorption by increasing the surface area. It reabsorbs water and solutes from filtrate and secretes substances into the it. So this is the general structure. The podocytes of the visceral layer. You can see a nephron with fenestrated and endothelium forming the filtering membrane. It gets filtered into the tubule, and where the structure differs in the proximal convoluted, we have microvilli and enriched with mitochondria with highly enfolded plasma membrane, whereas the distal convoluted tubule doesn't have much microvilli. The loop of Henle is thin segment flattened cells, and so the structure differs in each of the tubule. The loop of Henle, which is an hairpin-shaped loop of the renal tubule. The proximal part is similar to the proximal convoluted tubule. The proximal part is followed by the thin segment of descending limb, which has a length of around 2 to 14 millimeter. It is lined by simple squamous cells, whereas the thick segment of the ascending limb, which is 12 mm length, is lined by cuboidal to columnar epithelium. So there are two types of nephrons. The one which is in the cortex is called as cortical nephrons. The one in the juxtamedullary is called as juxtamedullary. The cortical nephrons have thin loop of Henle whereas the juxtamedullary has long loop of inlet almost dipping into the medulla. Only 85% of it is cortical, whereas 15% is juxtamedullary. The cortical is concerned with the excretion of urine, whereas juxtamedullary is concerned with concentration of urine by countercurrent mechanism. It happens in the medulla by the juxtamedullary nephron. The distal cannulated tubule, which is about around 5 mm in length, is lined by the cuboidal cells with few microvilli that function more in secretion than reabsorption. So we have the collecting duct, which is around 20 mm in length, which has two types of cells, the intercalated cells and the principal cells. The intercalated cells are the cuboidal cells with microvilli. They function in maintaining the acid-base balance, whereas the principal cells maintain the body water and salt balance. As you all know, as I told you, the length of a nephron is between 45 to 65 mm. The types of nephrons which I told you already is the cortical nephrons are 85%, whereas the juxtamedullary, they are located in between the junction of the cortex and medulla, have long loop of inlay, they deeply invade the medulla, have extensive thin segments, are involved in production of concentrated. The cortical nephrons are supplied by peritubular arteries, whereas the blood vessels in for the juxtamedullary nephron are called as vasa recta. So you can see the two types. The cortical nephron is short loop of inlay, which is covered by the peritubular arteries, whereas the juxtamedullary, which is present between the junction of between cortex and medulla, has long loop of inlay almost dipping into the medulla to maintain the concentration of urine by countercurrent mechanism. So finally we have JG apparatus, which is very important in synthetic and regulation. So you will be knowing the juxtamedullary apparatus, which is formed by whenever the distal convoluted tubule goes up and touches the afferent arteriole. That means it comes in contact with the afferent arteriole. The distal tubule and the afferent arteriole undergo changes in the formation of cells. The afferent arteriole forms specialized myoepithelial cells called as JG cells. They secrete renin, whereas the distal convoluted tubule forms cuboidal cells called as macula densa, which are the sodium sensors. This plays a role in regulation the GFR, forming the tubulo-globular feedback and glomerular balance. So very important is juxtamedullary. So macula densa is a sodium sensor, whereas JG cells is formed by specialized myoepithelial cells of afferent arteriole, and they secrete renin. So that's all about the functions of kidney. In today's class we came to know what are the excretory and non-excretory, the detailed functional anatomy of structure and function of the nephron, and different functions and types of the nephron, which are cortical and juxtamedullary nephron. So the assignment topic is draw a label diagram of nephron with its functions. Second question is, what are the functions of kidney? Both excretory functions and non-excretory functions. Draw a label diagram of juxtaglomerular apparatus with their functions. Thank you.